Through our preclinical research collaboration with Memorial Sloan Kettering, some of the exciting data that they presented at the triple meeting is really focused around the mechanism of action for seribantumab, which has not been previously characterized extensively in NRG1 fusion cell lines. So specifically within NRG1 fusion cell lines, the Memorial Sloan Kettering team was able to show that seribantumab not only has the ability to block HER3 ligand dependent activation, but they were also able to show that seribantumab has this unique ability to shut down phosphorylation signaling through the entire ERB signaling axis. So not only was seribantumab in these NRG1 fusion cell lines able to block phosphorylation of HER3, but it also has the ability to block the phosphorylation of EGFR, HER2, and HER4, which leads to complete shutdown of downstream signaling pathways of PI3K-AKT, as evidenced by phospho-AKT levels, as well as the MAP kinase pathway, as evidenced by phospho-ERK1/2.
And it's gratifying to see the hypothesis play out in that when we administer seribantumab, that we can see inhibition of the HER3 and we see inhibition of the downstream signaling of all the ERB family members. And this is really the first model or models that have demonstrated that. So that's an exciting move forward for us.
Most notably, the data of greatest interest is the data that Memorial Sloan Kettering was able to generate in the primary patient-derived PDX models, showing that seribantumab at clinically achievable doses is superior to that of afatinib at the clinically equivalent dose of 5 mg per kg, which equates out to the recommended and commercially approved dose of afatinib at 40 milligrams daily.
The other important thing I think that came out of those studies is that those responses in the animals were also very durable, and we didn't really see them escaping. Even after we stopped the drug. So I think those are very important findings. And we didn't see this with a non-selective ERB inhibitor like afatinib.
In the phase 2 Crestone study, we're dosing at 3 grams of seribantumab, which based upon the preclinical modeling is well above the allometric dosing range where we'd expect to see efficacy, which would give us a high degree of confidence that regardless of fusion partner and regardless of tumor type, that we should see efficacy.